Hi, I'm Paul Graywall with Sick Venture Studio, and we're here with a panel today to talk about emerging trends in oncology. And I see a lot of familiar faces, a lot of folks from oncology, literally everybody here, and a wonderful panel here. I'll let them introduce themselves, starting with Chris in a second. And when you do that, let's try to maybe say something brief about what yourself, as well as like your current developments in cancer, right? Or what you're doing to um, promote whatever you're promoting. Sure. My background is I'm trained. I did training in immunology, software engineering. I built companies. I've helped launch over 250 startups. We have and currently Sick Venture Studio is a fund and a studio that incubates Stanford spinouts in oncology, therapeutics, diagnostics, AI, precision oncology. Chris.
Okay, I'm Dr. Chris Apfel, made in Germany over half a century ago, which you can actually recognize from both my accent and my gray hair. I've asked way too many questions, so they kicked me out and I had to flee to the US. Being originally an anesthesiologist intensivist, have done a lot of clinical research, published over 100 papers, have developed a clinical prediction model that's now standard of care until my dad was diagnosed with lung cancer. And I realized that there are some limitations with genomic testing that can be overcome by really testing the original patient's tumor. So we founded Sage Medic. That was not out of UCSF. I actually left the clinical department there and founded this together with my wife. And we have now been able to develop an assay where we can take a patient's live tissue, create hundreds of microtumor replicates that reflects the tumor heterogeneity and the tumor microenvironment. And then we can drug them and see until they are really drunk. And then we can, after a week, we can actually see how they're responding to it. And then our latest news is that we got a patent issued that actually allows us to multiplex cytotoxicity and antiproliferation, because some drugs are actually more antiproliferative and some drugs are more cytotoxic. We can differentiate between those, which one is actually important for tumor response. The other one is to increase progression-free survival. And we can test it and have a test result for the patient within 7 to 10 days. And that's a big deal. We are CLIA certified, fully accredited by CMS. It's an LDT, a laboratory developed test, and which apparently doesn't need FDA approval. And it's now available. So if any of you is planning to get cancer, just talk to me.
Hey everybody, I'm Julianne Averill, and my background is I'm the finance person on this panel. So I work for a professional services company, Danforth Advisors, and what we help companies do is they think about if they have a strategy, they help turn it into action. And so that's action on the financial operations side, the clinical side, and then even market access. And so my companies, they tend to take their products and they can do anything from have an AI platform that focuses on target discovery and then moves into the clinic, or they're even focused on figuring out what does that precision diagnostic look like. Or we even have companies right now that are in the telemedicine and longevity space, figuring out how do we actually help the patients in times of need. And how I got started in this space was actually when I was 10, I worked with my parents in their medical clinic helping on the ops site. So I actually was helping my parents figure out how do you do the doctor's clinic side. And then where we are today, I also serve as an audit committee chair for Cure Search, where we help find cures for children's cancer fast.
Hi everyone, I'm Christabel. I'm a new faculty at Stanford in the Department of Rad-Onc. My expertise is in cancer immunology and the tumor microenvironment, and my goal is to develop therapeutic strategies, especially immune therapies to target, innate immunity, so inflammation, fibrosis, and the crosstalk between cancer cells and the innate immune milieu. We're spinning out our company, hopefully, within the next 6 months from Stanford, that we've developed a recycling antibody to treat ovarian tumors as our first indication. Happy to be here and answer any questions and look for opportunities to work together.
That's a hard act to follow. I'm Charles Keller. I'm a pediatrician and a children's cancer doctor, scientific director of CCTDI, a Children's Cancer Therapy Development Institute, a nonprofit biotech spinout of Oregon Health and Science University. So we do basic science. So I've been NIH funded for 23 years. We have a Cancer Research UK Cancer Grant Challenge grant actively too. And the work we do is related to antibody drug conjugates, small molecules, and protein degraders, which includes the work we're doing with Nurix Therapeutics in Mission Bay. They're really heroes of pediatric cancer, trying to develop 5 new drugs for childhood cancers in markets where there might only be 400 to 450 kids per year in the United States with those cancers.
Okay, thank you. So maybe I'll start with a question coming back to Chris Opfel over here. And so you have a platform and a new platform to test and to do testing. And then what do you think, can you talk a little bit about further developments that you see beyond what you're currently doing, what you see out in the market, and where you see the direction of going? For example, if we all started to, you started to test everybody the similar way.
So I think what is clear is that with our platform, we can test tumor tissue independent of its original primary origin and independent of the presumed mechanism of action. And so that is applicable for a wide range of solid tumors, especially in the late stage, when they also have mutated and have developed their own sensitivity and resistance profile, and you cannot know this until you have tested it. And it's also interesting especially for rare cancers, so pediatric cancers, but also the whole field of rare cancers where there are often no large clinical trials. That's actually where the Sage Oncotest is useful. The other part is, since drug development was mentioned, we can also test drugs that are planned to be taken into the phase 1. So instead of a PDX model, and we are basically faster, cheaper, and better than a PDX model because we have the original patient's tissue. We culture this in the own patient serum as well, and we can then distinguish between drugs that are more likely to work on primary tissues, and that's actually the important part, to work on primary tissues. One often says, well, a mouse model doesn't translate to the human, or a petri dish is even further away. If you put a cell line into a mouse, you still have the result of a cell line. And so it's actually, the key is what's the primary tissues that you're looking at, and that's actually SageMatic's core characteristic, to use patients' fresh, original tumor tissue to make a prediction.
Okay, Chris. So a lot of people have tried this. There's like 20 companies on the market, 10%. Why are you succeeding?
Well, the previous companies, so we have actually looked into this very carefully before we started Sage. And so first of all, there is the assumption on oncologists, it has never worked. Actually, if you look at the data, and we have done our own meta-analysis, there's actually an 80% sensitivity and specificity to make a prediction. And one can actually double the patient's chance of a tumor response by understanding what is the therapy that's more likely to work, number one. Number two, there were other companies around that, for example, were growing tumors. So the clonogenic assay and organoids, in a way, are part of this group, which is heavily funded by the NIH for many decades. But organoids need time to grow. They need weeks and sometimes months to grow. And low-grade tumors often don't grow. So then you don't have a result. And so the advantage of the Sage Oncotest is we actually are not an organoid model. We take the original patient's tumor and are not dependent on the tumor to grow. That's one of the advantages. then tumor microenvironment, then we can actually measure cytotoxicity and anti-proliferation. So there are many things that only we can do.
Okay, wonderful. Okay, you want to talk about climate? You want to talk a little bit about sort of the finance side and emerging trends in finance and so forth, right?
Sure.
Okay.
Yeah. So the emergent trends in finance. So right now, for example, with companies, what we're seeing is, as we've talked about earlier, there's a lot of innovation and there's a lot going on with these new and emerging technologies. However, as we try and figure out how do we fund these companies and fund these technologies and bring them to market, it's been a bit challenging. So what we're finding is early stage companies to late stage companies are trying to figure out, okay, what do we do now? For example, we're seeing changes and struggles with the NIH, governments, and what do we figure out? And so from that perspective, I'm starting to see a lot of my organizations as well as additional organizations move towards creative strategies. And what those creative strategies are is you align your product and your fundraising strategy with basically the funder's need, as well as, like we mentioned earlier, the capital efficiency. And what I mean by funding need is you think about partnering with investors where they actually have a focus on patient outcomes, as well as some kind of alignment around either location or product. And so, for example, at Cure Search, for example, since the focus is on finding cures for children's cancers fast, if your particular product can be converted to a therapeutic for children's cancer, then you actually have the ability to partner with them on grant funding there. Or additionally, there's funding opportunities with the state of Texas. There's also international funding opportunities with Saudi Arabia, as well as international with Italy. And there's different opportunities that can be found as long as you're willing to have your organization partner with those organizations and those countries as well as those states.
Okay, so lots of different new emerging finance opportunities. Okay.
Yep.
All right, so what's emerging at Stanford?
A lot. I think I don't want to comment on every innovation from Stanford, but so I'm a huge fan of immune therapy because I think we focus so much on just targeting cancer cells, and it's not just that, right? You have the entire, there's a crosstalk between your mesothelial cells and your cancer cells and your macrophages, and developing therapies that can efficiently target this crosstalk, I think, is the future, and a lot of labs at Stanford are working on this. Also, I think chemoresistance is a big issue, and as Chris's company is working on developing tools to test what are the best therapeutic strategies. I think developing combination therapies, which are immune therapies that can overcome chemoresistance, is the future, right? And that's what we're working on, and I truly believe if the science is sound and robust, speaking like an academic.
Can you double click on that? So what does that look like when you're combining those two? What does that therapeutic approach look like?
Yeah, so for example, what we're working on is ovarian cancer has the biggest issue is cisplatin resistance for patients, right? And where our next indication is HCC and other solid tumors. But we want to prime cold tumors, to make them into hot tumors where they're easy to target. So we're not gonna take away, chemotherapy, but what we want is to give our tumors immune therapy. So the therapeutic we're working on, we're trying to block the crosstalk between mesothelial cells and the cancer cells. And so when we block this, we chemosensitize our cancer cells to cisplatin therapy. So the patients would be given a much lower dose, so cytotoxicity won't be a problem, and you're priming the patient's immune system, right? And so in a way, this is, not just a short remedy, but something that can improve the quality of life of the patient. Not just they're not just surviving, but also having a good life 10 years later. Yeah, so that's.
So is that combining immunotherapy and oncology? Is that non-oncology?
Yes, so that's immune oncology, right? Because we're trying to elicit a response from the immune cells. So we're blocking this interaction between in our case, mesothelial cells and cancer cells. But we're also developing other therapies that can prime macrophages, for example, to attack, your cancer cells. So the answer here is, can we develop methods, right, which can prime your immune system, inhibit any interactions with the cancer cells, where the immune system is, like tumor microenvironment tells your cancer cells, right, like grow. And so you want to block that interaction. So it's kind of like an orchestra, right? Like I think of solid tumors like a, very well functioning orchestra. And, you want to block this signaling. And the way to do it is using, in my opinion, my humble opinion, antibodies. And right now, I think recycling antibodies are the future. There was one that was approved. And the advantage of a recycling antibody over monoclonal antibodies is that they bind to, your antigen, and they degrade it actively. So when you have a protein that is highly expressed in your serum, you're not just having your monoclonal antibody bind to it and stay there. You're actively degrading this protein. And so, there's a high, for proteins which have high turnover rate, you're getting rid of them in like real time. So yeah. So
Okay, wonderful. Okay, next question. Can we double click? You mentioned a little bit about ADCs and small molecules and your approach. Can you talk about a little bit about what's emerging and why you chose those modalities and what else is out there?
Sure. Well, so childhood cancer is the beneficiary of a lot of wonderful people in pharma and biopharma who are looking for simple single gene diseases where they can get proof of concept that their drugs work. And so I think one of the really neat advances these days, and especially since we're here in Palo Alto, the Silicon Valley, I come from the Silicon Forest, just outside of Portland, Oregon, where there are R&D facilities for Intel. So computational drug design. And so, we all hear about machine learning and artificial intelligence, and AlphaFold 3 is real. It's fabulous. You can do a lot of prediction of protein structures. You can do a lot of drug docking. You can make those drug dockings not just static, but you can heat and cool those computationally and find new drugs. Lawrence Livermore National Labs with BioBridge have developed a KRAS inhibitor drug, which has come to the clinic and is very exciting, just showing the power of computation. And sometimes those computations aren't machine learning or artificial intelligence. Sometimes they're just because we have big computers and we can do actual molecular modeling, even quantum mechanics, which is the side gig of Lawrence Livermore. So computational drug design is exciting, and it's here, and we're able to drug now not just enzymes, but curiously, transcription factors. So that's something exciting to talk about. Some of the other things that are going on, well, you can take those small molecules that you might be able to dock to a protein, and you take them right to the recycling bin of the cell. So protein degraders, molecular glues. If you remember from the 1960s, for people who are about my age, a number of people, their moms took a medicine called thalidomide, and so they have quite short arms. But that we've come to figure out was a molecular glue, and so you can use the same technology, the same principles, to take a driving cancer protein and put it right in the recycling bin. And so that's what Nurix Therapeutics is doing, and they've had some really nice public data on their BTK protein degrader. And so our Venus has an estrogen receptor degrader that they did a phase 3 trial, which in a subpopulation had nice activity. So take a look at that, both publicly traded companies. I'm not saying anything other than the public information. And then I think the really neat thing, because it's kind of having a moment, are antibody drug conjugates, and those drugs might be chemotherapy. We finally figured out how to link an antibody to a drug payload with a linker that doesn't fall apart. But those payloads, they can be chemotherapies, things very traditional like topoisomerase inhibitors, or things that are very novel, like DNAs that interfere with the expression of genes in things like myotonic dystrophy, which is a disease that certainly needs a lifelong treatment. Might be rare, but if you look at the multiples of the patients taking that treatment every week for the entirety of their life, sort of the Genzyme model, there's a lot of money to be made by investors who will pick a rare disease and aim to cure it.
Okay, wonderful. Thank you. So, Chris, question, ovarian cancer came up earlier. And Chris, you have a use case right for ovarian cancer. Can you tell us talk a little bit about that use case?
Sure. We have dozens of use cases. Actually, we published at the AACR, presented actually our novel multiplexed cytotoxic and antiproliferative assay, where we actually were probably the first one who actually found that there are certain drugs that really inhibit the growth without really killing the tumor. And that's not necessarily the same information. So PARP inhibitors, which are currently the big cost burden in the ovarian cancer space, are actually primarily extending progression-free survival, usually sometimes by years. So it's a real game changer, but only for a certain number of patients. And you don't see this in a cytotoxicity assay, which other companies are always doing, but only in our antiproliferation assay. There you see actually big differentiations in terms of PARP inhibitors. And the other drugs that actually inhibit growth and proliferation are the taxanes. So we can actually see whether there is actually another drug, an alternative drug, that's actually a generic, inexpensive paclitaxel, docetaxel, that may actually be more effective than a PARP inhibitor that costs $100,000 or $200,000. So that's kind of one in general observation. Interestingly, we had yesterday a conversation with a patient who presented with ascites as first symptom, so fluid in the belly. We actually got the ascites. There was quite a number of cells in there. We were actually identifying this. We saw very high proliferation. So this patient should have, in my mind, gone to surgery as quickly as possible. And the oncologist elected to go for the neoadjuvant therapy. So the idea is you give some therapy first to shrink the tumor so that you have better conditions. That's been quite popular in the United States, not as much in Europe. And I said, okay, so there is some sensitivity to cisplatin, but not to paclitaxel. If after 2 or 3 cycles it's not really responding well, then actually one should actually move swiftly towards the surgery, and then the other PARP inhibitors, which had showed some effectiveness. Got a call yesterday. The patient had 5 cycles of neoadjuvant therapy. The CA-125 didn't go as down as we would have liked to. So she had really 5 cycles of therapy that wasn't promising in the first place. And this tumor, it was a tumor that has very high proliferated quite strongly. So that's a case where the oncologist had that information, but actually didn't really act on it, and wasn't really, in my mind, on top of things to say, okay, the assay has shown this patient's tumor is aggressively growing. It may have some sensitivity to cisplatin, probably not to paclitaxel. Let's try the neoadjuvant, but if the neoadjuvant doesn't work, then rather than making the patient suffering all the side effects, let's actually then make sure this patient gets to surgery and maximize the outcome.
Okay. All right. So we only have 5 minutes left, so we'll go for a couple of quick comments left. And do we have a Q&A after that? No. Okay. So
Yeah, so I think I would just add too with the comments so far is the other thing I'm really excited about with the future of technology is this intersection of therapeutics and diagnostics. Like, for example, I have a company coming out of the UK that is blending their diagnostics engine, which is an AI platform like we were talking about earlier, and they're using the diagnostics from the AI side to identify potential tissue, take that through the drug development phase, and then use the data from the drug development side to then feed the
are you
diagnostic.
talking about biomarker-driven drug discovery?
It is, yes. And so it's been really interesting to see all of that. And so what I'm excited about is as these technologies continue to evolve, like seeing the integration and intersection of our different areas.
Wonderful. Christabel?
Yeah, I think there's 3 things we need to focus on, right, to overcome challenging diseases. So one is early diagnostics, because people don't know if they're sick, right? Second is better therapies, and third is effective testing systems that can recapitulate the tumor microenvironment or the disease. And I'm hoping that all of us here in the room are working towards each of these, and we can combine them together to solve the big problem of, overcoming these diseases.
Thank you. Sure.
So as news you can use, and as a plug and an advocate of kids with cancer, it's kind of amazing the resources that are available to startups, but also investors when you think about the companies that you support. So in childhood cancer, if you enroll in a national clinical trial, they'll check for any common mutations. And they'll return those results not just to the doctor, but to the patient within 6 days. They've open sourced all the information that's available for childhood cancer trials through the University of Chicago program. And you can just sign up with an email account. You don't have to have an affiliation, just promise to use the data properly. Foundation Medicine's outsourced all the mutational data from kids that have relapsed from childhood cancers, and Champions Oncology makes all of their model information for the patients that have had new diagnosis or relapsed patient-derived xenograft models available for free. They'll even share the mice. So it's just a wonderful place where if you need proof of concept and you need consistent data on a national level, you can go to these resources and you can prove out your platform and you can show your value proposition.
Okay, wonderful. And speaking of early detection, I'd like to say the Grail folks are here somewhere, right? So just want to thank for being out there, and then you guys should really consider taking that test. I mean, it's probably the top test in the world right now for early detection. Gallery over here, and then also I'd like to say thank you to Roger for putting all this together, and Ari and Brad and folks. All right, thank you.
Now we're going to shift from developments in cancer care to more the personalized precision approach that we're seeing more often these days. And here comes Marty. Come on up, Marty. You're just in time.
And thank you, Roger, and welcome to the panel. I wish someone had told me I can't use Apple Pay with an American Express card on the parking app, or I would have been on time. So I'm a computer scientist and an entrepreneur and a visionary, and was lucky enough to be in Silicon Valley during this amazing golden age, which continues to this day. And robots I built in the 1970s are in the Smithsonian and the Computer History Museum. I started the first company that was focused specifically on electronic commerce. We hired Mark Andreessen and brought him to the Valley. He spun Netscape out of the company, which was called Enterprise Integration Technologies. And we started the association, the alliance, that made e-commerce possible by taking all the small companies, and Netscape at the time had like 20 people at it. Amazon had even less. We brought them together so they could work with each other. And so we could bring in the FedEx, the banking, the shipping companies, and so forth, in order to be able to make all of commerce happen end to end. And that's kind of the situation we're in today with precision oncology. We have a lot of the pieces, they need to be connected. In 1998, I was diagnosed with melanoma metastatic to liver and given 9 months to live. That was before any of the targeted therapies and checkpoint inhibitors, which were developed in melanoma simply because there was nothing else in that disease. It was really deadly. My life was saved by a clinical trial that failed. That's where luck came into it. But when I survived, I kind of pivoted to a much more significant undertaking, which was trying to use my skills as a computer scientist and an entrepreneur to make a difference. And I started a company called Collabrix in 2008, which was the first company to offer precision oncology navigation as a service. And we were using RNA-seq in those days, using Effie chips. 3 years later, I spun out Cancer Commons, which is a nonprofit that helps patients who have exhausted the standard of care navigate the cancer maze. And Over the years, we've helped over 10,000 patients. And I'm very, very proud of that. But it's a drop in the bucket compared to what's needed. The need is kind of 10,000 patients a month. And that was beyond my managerial skills. So I'm very pleased to say that as of last Monday, I gave up being the CEO of Cancer Commons. And I brought in a really experienced leader who will be here this afternoon, and I hope you'll all take the time to meet him. That's Cliff Reed, who, among other things, did Complete Genomics, took it public, and more recently Travera, which is a functional testing company. I'm going to run Cancer Commons Labs, which is kind of Marty's sandbox, to be able to work with all of you. Cliff has a very ambitious goal. He's aiming for 100,000 patients a year, and he aims to get there by using AI to curate all of the advanced treatment options and match patients to them, and then track the patients to see what works. It's something I aspired to do, and it was beyond my pay grade, but Cliff's going to do it. But we can't do it without your help. And the reason I say that is because our goal is not just to curate the options that are out there, but to create new options. And that's where all you guys who are investors and startups come in. And I want to work with you all in labs in order to be able to engage you, bring you to clinic much faster than you're able to do. And I'm sorry. Okay, could everyone hear me now? All right, and be able to accelerate your progress, give you the data that you need to be able to train and validate your algorithms, for example, but most importantly, bring these things to the patients much sooner. So at this point, I would like to introduce the panelists and simply say that precision oncology, personalized medicine, is really something that's needed on multiple levels. Obviously, the patients need it, but so do the people developing drugs. Because, as you all know too well, developing a new cancer therapy, or a new test for that matter, is extremely expensive. It's time-consuming. It's risky. You know, the number I hear, and I don't understand how this number happens, is an average of 2.6 is the latest number, $billion to bring a new cancer drug to market over more than a decade. And the trials where most of this money is spent are focused on population statistics. So whether the drug will do any good for a given patient is a crapshoot. So I'm going to turn it over to you guys now, and we'll have a discussion.
Hello to everybody. My name is Brad Niles. I'm CEO of Arise Precision Medicine. We're a Davis-based startup working in the siRNA field. And so where we got our start is having family that had gone through their cancer journey, seeing the side effects that come from a lot of these toxic therapies, and really a goal to create something better in which the patients could eliminate their cancer without killing off all their normal healthy cells and all the side effects that come along with all that. And so really to get to a side effect free type of cancer therapy, we needed to go directly at the source of the cancer. And we were fortunate at the time to identify some really exciting research that was coming out of San Diego with an individual that had identified one of the early changes in cancerization of the cell, epigenetic regulators. So long before any of these downstream mutations occur, you have changes in gene expression that drive a lot of these pro-signaling pathways. And so we picked up where this investigator had left off, developed a therapeutic approach. We identified siRNAs as the best modality to go after this particular target in order to get some uniqueness in between a truncated version of the gene that was in the cancer cell versus the normal healthy tumor suppressor version. And so we spent the last couple of years working on a drug development project. We are in the preclinical stage. We've taken our siRNAs from in vitro, paired it with a targeted drug delivery system. It's a calcium phosphate nanoparticle. Allows us to get specifically down to the side of the tumor, get better uptake into the cell, and allow these siRNAs to do their mode of action.
What's the time scale? When's that come into clinic?
So we hope to get our IND application done in about the next 2 years and start shortly thereafter.
How long have you been working on it?
We've been working on it about 5 years at this point.
2.5 billion.
Hi, everyone. I'm Laura Shea, the founder and CEO of Tipping Point Biosciences. We're a UCSF spinout out of Dr. Geeta Narlikar's lab at UCSF, who's my co-founder. And so we're also an epigenetic therapeutic company. And so one of the biggest problems in targeting epigenetics is that most people have targeted single epigenetic factors, but these factors have roles both in normal cells and in disease cells. And so in order to get better specificity, we came up with a different idea and technology to really target these epigenetics and reprogram the epigenome in disease in a very specific way. So we developed a platform technology that recapitulates the epigenetic proteins actually in the context of how they function on chromatin. And so the key is that a lot of the folding and the interfaces that are now accessible when they're in the right context in the on the genome is actually very different in disease versus healthy cells. So we're trying to drug these unique interfaces. So this is something that, AlphaFold can't model yet because we don't have the data. So if we can generate these networks of interactions to understand the new types of interfaces that are unique in disease, we think we can get better therapeutics and more personalized medicine for specific networks that specific epigenetic networks that go awry in disease and in cancer.
This is personalized medicine on a long time scale, isn't it?
Yes.
We're going to get back to more specifics in a second.
I am Amy DeGolier. I am the founder and CEO of Represent. And we are building the missing human signal that's been lacking in precision medicine. Essentially, precision medicine has made incredible advances, but when you're using the genomics, you're using the imaging, you're using the labs, you're using what's in the EHR to train out these AI models, there's a really crucial piece that we've overlooked, which is the 99% of the time that patients are apart from the clinic. And it's how the patient is experiencing their treatment. It's how the therapy is working within the patient itself. And the challenge is you may, put together the best treatment for this individual patient and they go home and they go septic because for that patient's individual biology or something that happened home, it all went awry. What we're doing is capturing this lived experience in a dense, frequent, structured way. We're applying ML to find the patterns within it, and over time using these signals to identify things like progression of disease, efficacy of treatment, and patient risk at large. In the first order, we've created a system that really helps patients personalize their own care so that they use our system deeply so that, they get better control of their own symptoms. Their doctor can better adapt their care. But in the long run, we're looking really to develop this whole layer of precision medicine to make the entire field more accurate.
And that's an example of just one of the many pieces of missing infrastructure that's needed in order to be able to bring precision medicine to market. So I think at this point, let's try to dive deeper into specifics. And I'm looking here for challenges that you faced in your companies and ways to overcome them, specifically around things like funding, regulatory processes. How can we make the whole clinical development process, clinical trials, much more efficient? And what other pieces of infrastructure are missing? For example, Cliff Reed, who had run a functional testing company, was stymied by the inability to be able to get specimens from community hospitals, or even from Stanford for that matter, within 24 hours so they were still fresh and he could do his testing on them. There's a lot to be done. So you guys, aside from whatever you're doing at the moment, are all very experienced. And make us smarter. Whoever.
Yeah. Well, I'll dive in on the clinical trial piece just for a second because that's actually where we've been leaning in very heavily recently. There's so many more toxic therapies coming to market, and it's wonderful to bring these therapies that are needed. But the challenge if you're a drug developer is, you need to move fast and you need to move fast safely. And if you get a high grade toxicity as part of your trial, the whole thing could be over, and this incredible treatment that's needed for so many patients could be lost. And so, as an organization, to really develop more signal and to drive volume, that's where we've leaned in to help trial developers, identify these AEs sooner, to lower these high grade toxicities, to make the treatment more tolerable. Because if we aren't able to get these drugs out to the patients that they might be relevant for, we're really stalling development in that long-term development piece that you mentioned. Overall, we're not making the progress we need. So in the first order, I think that's the first application that we're using for this precise and personalized element. But over time, it's then, of course, to identify which patients it may be suited for in the first place.
Anyone else want to comment on trials?
Well, I mean, kind of related is I think that as a drug developer, I think that it's really important to understand why drugs fail in the clinic. And unfortunately, a lot of the clinical trial data is not accessible, right? And so I think that makes it more challenging. If we could be more open source and share some of this data, it would be a lot more helpful in terms of understanding some of the barriers that we need to get over early in development, before making similar mistakes when we actually run the clinical trials.
Yeah, wholeheartedly agree with that, and wish there was a better system. Maybe somebody can figure that out. But when we started our drug development process, we tried to work backwards from clinical trials with the idea, we know what we want it to look like when it gets to the clinic. How do we reverse engineer that down to the most basic levels? And the primary function or the primary point to consider there was failure due to off-target toxicities. And so an siRNA approach we believed could avoid a lot of those potential pitfalls with off target. And sRNAs and RNAs in general have been kind of maligned over the years. One of the early issues with sRNAs was an off target in terms of the complementary sequence. Where sRNAs have really advanced since their discoveries was in the delivery. And so our major focus was not only to design the most effective and most on-target sRNA, but actually be able to get it specifically to the site of the cancer. And if you're not getting it taken up into normal healthy cells, you're not going to have those side effects. And so our major focus was in nanoparticles, not only just like the LNPs that a lot of us are familiar with that often go to the liver. How can we get beyond the liver? How can we talk specifically to this interesting thing's going still on? How we can get it specifically to the site of the tumor? Cancer is the maybe it's a battery. Lung cancer is our cancer of interest. Because of a huge market in those patients that have the defect that our drug goes after. So if we can select patients that will respond to the treatment, if we can get the treatment in high enough levels down to the site of the tumor and only get uptake into those tumor cells, we can avoid a lot of the failures that others have dealt with.
All right, since you challenged in the first thing, anyone has a better idea, I want to throw out a better idea.
Excellent.
It's very provocative. It's a complete paradigm shift. It's flipping the goal of trials from approving drugs to curing patients. And I'm thinking that this could address a lot of the issues that we're facing, but with a very big caveat, which is that this is really not aimed at first-in-human drugs, because those should be done in a phase 1 unit for sure. But if you think about starting with the patient right? And we at Cancer Commons, all of our patients have exhausted. They don't come to us till they've exhausted all the standard care, right? And then we have to find something for them. And the idea of starting with one patient and really doing precision oncology on that patient, working them up, doing all the omics, all the functional testing that you can, doing computational and systems biology, even digital twin, to try to do the best job that you can. Every patient is going to be different with advanced cancer, right? There is no trial. You can't, right? It's all about trying to find what the optimal combination of therapies is. And it almost certainly, I make a very provocative thing. No one knows how to use the thousands of drugs that are available in an optimal way for a given patient, right? That's precision medicine. And so if you do something and it works, what you're really perfecting as much as whatever combination of drugs is the technologies that you're using in the core of all the people out there who are developing tests and services to help eliminate the guesswork, right? So you get this patient and if the patient doesn't respond, you get another shot on goal. You try something else for that patient, right? But you're testing combinations of things, which is not normally done because there's too many of them in a trial and they want to get a pure signal on something. So you're curing the patient. There's no inclusion, exclusion, no randomization or travel required. Everything is focused on this patient, right? And then for you investors, you get to see if it works on one patient. That doesn't tell you anything, but it tells you one thing very important, which is that there's a there. And, the scientific risk suddenly goes much lower. And also, the market risk goes lower because this is something that the top physicians that we work with wanted to use for this patient. Eliminate market risk, eliminate scientific risk, and be able to then buy another vowel, so to speak. Invest a little bit of money to try it on a handful of more patients, whatever it is, the technique and the therapies, right? So you've made a very small bet, and you can make a large number of small bets for a given set of bucks. I think this is a better way to do clinical development and investment by flipping the paradigm. And I would really appreciate constructive feedback or even destructive feedback on this radical idea, because I'm intent on doing it and doing it commercially with many of you. And the audience can chime in here if someone has something to say.
Not a criticism, but more of a question. How do you manage the side effects that come from each one of those drugs when you're, technically stacking side effects?
I'll take that one, Brad.
There she goes.
We just don't happen to have an expert on that.
We just do. And I would say, to expand, where this idea is going for you, Marty, I think the other element to keep in mind here is the access, how we don't just do this in a small area. This is precision medicine, but it doesn't have to be for a precise population. And I think that's how our complement is really helpful. If we can start to create almost a symptom omics, if we want to put an omics on that, and we can start to use these signals so that we can push this out to the edges of society, it becomes not just let's find the signal in the center, but let's push this out as far as we can. And I think, Brad, your point is, how do you do that with the symptoms? You need to manage it in a different
There's
way.
even an opportunity to do science in real time, right? All these scientists who do cancer research, and many of whom have never seen a patient in their lifetime, right? Get them to weigh in on the initial treatment, and when something is not working, either side effects or the effectiveness, right? To get another shot on goal, a knowledge turn, I guess. The patient's getting another shot on goal. And you can do 3 or 4 of those a year, as opposed to locking someone into a trial protocol for multiple years. Let's do something different. This is Silicon Valley. I'm ready. And I don't have a lot of time to do it either. Probably in the next decade, it's got to get done, guys. All right. So then the next question, we're talking about incremental ways of financing, place lots of small bets, double down on the winners. Any other ideas? Financing, all these companies that I work with that are developing therapies are really struggling at the moment. because the funds are drying up. And what can we do to help those companies? Anyone in the audience? In back? Not a problem. All right.
You know, that's pausing. A lot of people don't realize the opportunity in crowdfinancing and crowdfunding, and this seems like an ideal place to do it because because I like to say crowdfunding is good for affinity investors. When you've got a product that consumers understand and they see the problem and the potential, I mean, this is a place that fits that, given that almost everybody knows somebody with cancer or has been affected by it. So that's one option, Marty. What is our panel? Someone in the back here. Malika, go ahead.
Someone earlier today asked me about free rounds and how you could support a company that someone else who's currently in the audience, they asked a little bit more about bridge rounds. So as you mentioned, as few companies are going through a tough funding environment at this time. And because they've already raised money from VC funds, they've gone through technical due diligence, they have strong leadership on their boards. They might have raised money from Bill and Melinda Gates Foundation, or I believe someone said that they're an ARC spin out. So yes, when you invest in a bridge round, you're coming in at a smaller check size than Bill and Melinda Gates are, but you are benefiting from the technical due diligence that they put the company through, or other big name investors. So just to give another example, I wouldn't totally call that crowdfunding, but I would still call it coming in with your own funds at a check size that's more realistic and doing it in a way that supports your own finances because the company has gone through technical due diligence as well as major commercial or social support.
Yes,
You get a micro VC, so you're right here. What do you had like the bar? Everybody liked it. Bob Bar, yes, Bob.
So are we hearing it?
All the companies in Silicon Valley for over 20 years. And since I'm a collector of data, what I'm thinking about in a group like this is I might put a group within LinkSV of just getting you all together connected to share ideas of what you've all learned, what you can contribute to each other, on who you might know who are investors, great advisors, great board members. I love all this stuff. My grandfather was the head of obstetrics at UC Berkeley, UC San Francisco in the 1900s. My dad was a doctor who basically was responsible for the labeling of prescription drugs signed by Ronald Reagan. My whole life is dedicated at this point to wanting to help advance things in medicine. But I'm an angel investor with a small amount of money. But I love the fact you guys are all doing things to make a difference. I can help you by maybe figuring out a way to get you to all share it together. I'm an expert at network.
well, let me add one thing. There is a radical idea here too, which is to tap a different source of capital, right? People who are affected by cancer. And there are, whether you do it through crowdfunding or you do it through ultra high net worth family offices, right? People who have donor advised funds or want to invest their capital. I'm going to be setting up a cancer impact fund within Cancer Commons to give people the option, if they don't feel like investing, like donating philanthropically, which has fueled all of our work to this point, to be able to provide dry powder. So when we see something that's working in the clinic, again, de-risked both scientifically and market-wise, to be able to place, to have dry powder to rapidly double down on these winners. It's a much better way to do it than I think the way the industry has traditionally done it. And I would love to collaborate with the investors out there to set this impact fund up. A lot of capital, trillions of dollars that could be capped. Yes?
So I'd like to double down on your idea about talking to real patients because I like to share an experience. And maybe others could learn from that too. So we are a nonprofit trying to help cancer patients, really more oncologists. We have an AI-based tool which helps oncologists to give them standard of care. A close friend of mine for the last 2 years, 3 years, got diagnosed with stage 4 pancreatic cancer. And he's been at UCSF. He goes back and forth. Just 2 weeks ago, he's kind of now given up all chemo. So he came and, he used our system, and the kind of feedback and the emotions that we got, when he used it, he said, I wish I had it, when I was getting my chemo, because there's so many questions that I can't ask the folks, in the nurses. And a lot of time I go for chemo and they said, I'm not ready for it. I come home. So my point is that this notion that you're saying you should talk directly to patients, at least we could, because we're not kind of giving them any drugs. It was more of a information tool. But I think that really hits home when what you say, because that interaction completely changed the things for us from day and night.
And I want to work with you. I want to work with all of you. I got the 1 minute flash. So anyone else have something that they're desperate to say? Please.
Well, I just want to share that I think this is a great idea to share experiences. We did a crowdfunding project while we were in the Cancer Hacker Lab. It's non-trivial. It's incredibly time-consuming. And I'd be happy to share all that experience. I think I can do it better next time. I've raised 6.5 million in grants. I didn't get the first one. So you're willing to learn. And the last thing I want to say is that I have been blessed with amazing investors that have supported us. And those angel investors are committed because of our mission and because we continue to update them. And by the way, you're talking about intelligent money and all of that jazz. They're pretty smart. They've done businesses. They have operational experiences. I talk to VCs with 0 operational experience. They are money allocators. They never worked in a company. So.
And 90% of the drugs that enter trials fail to become products. So,
Hey,
go
Roger.
figure.
I'll just, Roger.
Okay. Last comment.
I'll just mention, for example, by using a keyword search in our system, if you typed in cancer, we'd come up with like 137 companies in the Bay Area with 20 years have been involved in cancer, and I have all the investors. So the point is I can figure out that and share with you who might be investing in something you care about by a keyword. That's it. That in itself is probably valuable.
Okay, and with that, I want to thank our panel. Personalized care. So our next panel is on artificial intelligence and cancer care. Our moderator is Ranjan Neg. He is a professor at Stanford. He is a venture capitalist. He's a former entrepreneur. And he has the highest VO2 max of anybody I know. So with that, Ranjan, welcome.
Thank you. Is this working? Great. Excellent. Oh, we're missing one person. Where's she gone?
Oh,
there she is. Kamudi. Excellent. It's so nice to be here. And I was asked to talk about artificial intelligence. So the grand vision of AI, just like we heard in the last panel, maybe you don't need to do any trials. You just do the computation, click the button, and it comes out with the answer. Right now, our GPT systems are as clever as a PhD in one topic. If you get to super intelligence, 2029, some people think, there's a PhD level intelligence in every topic. And maybe we need that to solve these difficult problems. Yes, so I'm a venture capitalist. I'm a teacher here at Stanford. Some of you may have taken my courses. I'm teaching AI for grant writing right now. And if you need money, that's free money. And an AI crash course. Roger was my student. Thank you very much. And maybe some of you were. But we've got some super panelists today, and I'm going to introduce them. And then we're going to go straight into discussion. So first of all, we've got Dr. Kamudi Bawe. Kamudi is a founder and principal at YOM Consulting Services. She helps small biotech companies establish end-to-end AI, ML, multi-omics translation biology approaches to both therapeutic and diagnostic development. She also founded YOM Wellness, where she combines her scientific expertise and integrative healing knowledge to help cancer patients navigate complex health choices for themselves. And Comedy holds a PhD in public health and has oncology research experience spanning more than 20 years. Then we have Felix. I think you've heard from Felix before. Felix is co-founder of NeoTela, an AI-powered cancer monitoring and surveillance, as well as personalized immunotherapies targeting liver through precision diagnostics and next-gen cell therapies. Felix is mentor for Stanford Care, as well as the Stanford Biodesign program. He exemplifies the mission of Stanford Biodesign, bring the world's most promising minds into one room to investigate, innovate a healthier world for all. And he distinguishes himself as an exceptionally dedicated fellow. Finally, we have Dr. Nabil Adam. Nabil is Professor Emeritus at Rutgers University and co-founder and CEO of Falcon, a startup focusing on AI applications in medicine and healthcare. His first startup, which focused on the pharmaceutical industry, was sold in 2008. So an entrepreneur at heart. So let me jump straight in and think about AI. And Roger sent me this article and said, 2 years ago when ChatGPT sort of started to pop up and AI, I think I've been in AI for 40 years at this point. My first system was in 1983. So people think it's just a few years old. But when it first came up in mainstream, where the most popular programming language switched from Python to English, lots of universities, including this one, were pretty nervous about using it on their students. And now, actually, quite a lot of universities now have said, well, actually, go and use it. Go and use it. But by the way, we expect bigger expectations. We expect higher quality output. But medical schools are still concerned. We've still got these things about, well, what if it prescribes the wrong thing? And at Stanford, there's an enterprise version of all this CYA language. Do not use it for any clinical practice. And yet, I've seen this blog post that the latest systems are now giving reasoning. It does get it wrong occasionally, but it tells you can tell why. Because it gives you the reasoning, tell you why it's going. Can AI be used in medicine? Who will go first? Let's start with Felix.
I teach medical students. I teach doctors all around the world, and AI in medicine is here to stay. There's no way of turning back. It's a question of how we're going to use it. It's a question of how we're going to adopt it. It's a question of how and who's going to pay for it. So, so AI is here to stay, and AI is meant to empower both patients and families, as well as clinicians and administrators on better outcomes for patient care. Where cancer is concerned, I think that cancer, it's a very sensitive topic as well. The question lies in, do I want to know? Do I not want to know? But I'm of the view it's better to know than not to know. So I'm trained in hepatology. So I will focus a bit more on liver cancer because it's a totally silent killer. But I will not talk a lot because I'm going to present later in the afternoon. But yes, to Roger, who is the veteran here, AI in medicine is here to stay and it's going to make greater impact.
So Felix says it's here to stay, but are people actually using it now? And if they are using it, what are they using it for? And if they're not, why not?
Okay, thank you. I'll speak to this a little bit. All of you must have seen the news that's made the splash recently about Microsoft and the MyDxO and how it's so much more efficient than some physicians who were asked to diagnose these cases. Now let's dive a little bit deeper into that news though, because news articles are flashy about the latest, but let's see what it actually did. What was behind the scenes in this particular tool, right? That's the latest and greatest and the forefront of what AI can do. It's the news articles are saying, oh, is it going to replace doctors one day? Now we have to remember what Microsoft built is a tool called an orchestrator, right? It's a multi-agentic system. I'm bringing in AI jargon. Those of you who are much more well-versed can, kind of challenge me on everything I say here. But multi-agentic system. So what the orchestrator does, it puts together 5 agents. An agent is a step beyond what most people are talking about, ChatGPT, Claude, Gemini, all of these. An agent not only passively gives you information in a nice, beautiful format, but it actually can do stuff, right? It can book appointments, it can do those things. So they've put together 5 of these, and now it's acting like a tumor board would or a group of doctors would. However, you have to remember that this is trained on, or it's rather using the LLMs that already have access to all the information that's out there, including the NEGM reports. Has such a system been created for a real world situation where where there are a number of false positives that could crop up, or there's other concerns? No. This is experimental. It's sensational at the moment, right? But there's a really long way to go in terms of ever replacing doctors. It would be more like a tool that doctors use. One caution there would be, I'm just going to put one comment here, which is probably talking to a question that maybe you were going to ask me later, but I'm going to say this. As we humans are using these more and more advanced tools, the tendency of the human brain tends to go towards a little bit of laziness. And that's something I think we have to be cautious about. I'll stop there.
Okay, excellent. Laziness. Does it make us lazy? I mean, I asked this question yesterday to a class. Does AI make us dumber? Or does it actually make us cleverer? We have these tools, so, and I think the response was if at our level, it makes us cleverer, but at teenager, it might make them dumber because they're lazy, right? They'll just take one answer. But if you're a doctor, you might double click. But okay, Professor Nabil. Okay, what do you think? What do you think is the sense of doctors? Are they skeptical or are they frightened? What word do you, or are they actually using it in secret? But what do you think? What do you think people are doing in medicine?
I think in terms of AI in medicine, that certainly is here to stay. And I think it's good to realize that, but also realize that it is an empowerment to patients and clinicians, but it has limitations, and we should work hard on addressing those limitations. So, for example, limitations in terms of quality data. A lot have spoken about it. The validation of the models is very important. And the skepticism is healthy. I think it keeps us all in our toes in terms of researchers and clinicians. So we want that to be there. But also, let's think why the skepticism, because clinicians need to be convinced and have some trust in the results that the AI models provide. That's why explaining the results and how it came about, whether it's right or wrong, is important. Whether the physician would be convinced or not, that's a different. But the ability to explain the results, how it arrived, how the model arrived, those results are really important. Another, it's actually interesting, by the way, there's a recent study that was published in JAMA just a few months ago that showed that most of the oncologists that were surveyed, most of them recommended that their patient would consent to the AI models used in their treatment plan. Okay. So that is
the patients would consent.
Exactly.
So when the patients were asked, they said, yes, go ahead.
Right, the patient would consent to the result. So that actually shows that there is skepticism, but also there is trust building from the physician and clinician part of the equation.
All right. So AI models, they have utility in the diagnostics and the drug discovery space. But usually the critique, I forget who, maybe it was Camus who said, the false positives, the false negatives. But humans make false positives and false negatives too, right? So I'm not sure who asked. Camus, maybe I'll ask you this. The false, if something has a lot of false positives or false negatives, is it still useful? Or do we say, well, actually it generates more questions, I have to spend another $300 on another test to double check and a follow-on test behind that. And before I know it, spent $4,000 and something that didn't really matter anyway. Or does it catch early my phase 1 cancer in pancreatic cancer?
I think, thank you for this question. It's extremely useful, as we're already
So
saying.
even if
because-
it
Yes, it's extremely useful. It's extremely useful as a starting point. I mean, I talked about this news right now and how it cannot replace doctors yet. It's just a starting point. Meanwhile, let me address another aspect of the question you asked me earlier. What are the uses? There are patients and patient groups that are using AI, like Brad mentioned earlier. And something about my background, which I hadn't put in the little blurb, is that I have had the honor of working with Marty Tenenbaum and Cancer Commons as well, working directly with patients and their families in the past. So that I've built upon through Young Wellness, where I use integrative healing as well as a component when I work with cancer patients and their families. So cancer patients and caregivers are using ChatGPT and just Claude or any of these LLMs that they have access to ask and have conversations about the questions that they cannot ask anyone in the times that they're not visiting with their doctor or their nurses. And so it's extremely useful, but with a word of caution, because the answer you're going to get depends on the question you ask, as we all know, as we've all dabbled with our own conversations with these LLMs, right? We're all learning that the answers you get depend on the questions you ask, depends on the data it's being fed as well. As much data as you give it, it's going to get better and better. So if you stop, it's kind of like the question being asked is it better to be half knowledgeable or zero, ignorance is bliss, or completely? So in the reality, none of us here is completely knowledgeable. All of us are at different levels, right? And so it's extremely useful as a starting point, is what
Yeah, so I mean, I think when I used to go to my doctor, she said, don't believe anything you read on the internet. Now she says, well, Ranjan, what do you think it is? Because she knows I've spent 2 hours researching it and we only have a 10-minute consultation. So is it going to be a discussion instead of a white coat, you just do what I tell you to do, where the doctor is actually expecting the patient to have more of a back and forth? What do you think, Nabil?
Yeah, I think that's an interesting point, though. It's also, and I think we're speaking about the agents. That's where maybe we can focus having the agents take care of the routine tasks that currently clinicians does and spend time on, and release her from those routine tasks and have her spend more quality time with the patient. Have the agents, have the AI capabilities and model prepare the history of that patient that I'm going to see in few minutes, and have that summary of the MRI, the different reports, all of that. Give me an overview, holistic view of the patient, the treatments, adverse events, comorbidities, and so on over the past several visits. Have all of that for me prepared so that when I see the patient, I don't have to spend time going through this and memorizing. Have that in front of me and maybe focus more on what is at hand at this point.
All right. Okay. So this is back and forth. We're thinking that these systems are not quite perfect, but we can accelerate the treatment and the discussion, maybe some more accuracy. But how do we get to this sort of holy grail, and when will we get it to it where it just tells us, it just gives us the right treatment? How do we make it AI more accurate, and it just gives us the right medicine, the personalized? Maybe Felix can comment. How do you make it better? Why can't we make it perfect?
I think that the future of healthcare would be even more precise, given, we've got very smart engineers, we've got computational biologists, we have got scientists, and all this, and so someone mentioned about digital twin, and I'm a big fan of, and the future is about digital twin. But the only thing is this is who's going to pay for it is. But I believe that will somehow play a part in where healthcare is going towards, in terms of precision medicine, in terms of personalized healthcare, in terms of innovation medicine, and in terms of where the future looks like. So back to your question, I think I would put my bet, given that I'm also a venture partner, I would put my bet that this is the golden age of healthcare, never seen and never to be repeated ever again. And I think that by probably 2035 or so, the dynamics of healthcare, both to the doctors, both to scientists, both to researchers, will be very, very different from what it is today. And the way how we teach medicine in the university will also change. I'm certain of.
All right. Okay. So no, I work on aging, which is like even grander than cancer's just a subset. So I think about almost the opposite of personalized medicine. Can you solve all diseases? Can you solve all cancers all at once? Which we've been telling people for years that cancer's not one thing. So the Chan Zuckerberg Initiative, when it first started, at least the news article I read, they said, I think it was UCSF and Stanford, and told the Zuckerberg family, said, look, we can cure all diseases by the turn of the century. Just give us 500 million. And they did give them 500 million. So with AI, let me ask each of you this. Turn of the century, 70 years from now, will all diseases be cured? And then we don't have to worry about the cost because no one will get ill. Yes or no, and how? Kamudi, first of all.
All right.
This is
me.
turn of 70 years.
All right. So I think absolutely it depends on all of us. We all, lay people, have to also be participatory in this. We already have the tools. People are wearing the Apple Watches, wearing all kinds of technology. The opening session today was about the wearables and the time that's spent between doctor visits on the daily, our daily routine, daily healthcare that we do for ourselves. So if we're all able to, in pockets, participate in collecting the data around that and turning that data into useful and meaningful information, then there's a lot of hope.
So that's the how, but it's yes or no. 70 years,
maybe,
no diseases.
maybe.
Maybe Nabil will be more.
Well, I'm not sure we'll be able to cure more. The idea also, if you look back, we didn't know of some of the diseases that we know now. So it is a discovery as we go along. So for sure, we'll not be able to you cure all the. But I think we have to take it not one step at a time, one patient at a time. Back to Marty's point, we have to think in terms of that specific patient and how they cure it, how AI can actually help that specific patient with the entire background, not only the medical, but also, I think it was the holistic view of that specific patient.
I have a slightly contrarian view, just if I could interject. The aging, people who work on aging research almost think the opposite. They say, well, yes, each patient is different. Yes, obviously. And yet, if we can actually compute every single possible pathway that goes through every single disease and see what the most common pathways are. Maybe we can get one thing that cures lots of diseases all at once, so-called. So the aging field is different. It's the opposite to oncology, usually traditional oncology. So you say no. I think I heard you say no, but we've got some ways to do it. So we've got maybe Felix, 70 years. Can we cure all diseases?
In this context of cancer, I am confident that we can cure cancer. Okay. With technology like CAR T cell, armor T, with immunotherapies, I am quite confident. The intellect scientists of the world, be it here from the US, out in UK, in Cambridge, coming together, putting our minds together, I believe, and I'm hopeful that we can cure cancer. And we can start off with some of the low-hanging problems before we go to the deeper problems. But at the same time, and this is what I want to emphasize, many a times people are not aware. So surveillance and monitoring is critical. Secondly, your lifestyle and your diet is also critical. So if I empower you with all the necessary information in the palm of your hands for you to look after yourself and that of your families, will that make a difference? I think it will.
All right, so Felix says yes for cancer. We didn't get an answer for the other one. And the how was smart people and behaving yourself. But I still like chocolate cake. That's my problem. Let's have some audience. Any comments or questions from the audience, because none of us know how to solve everything. Gentleman over there on the right. Left.
My high school. Anyhow, so we're pretty bright kids, and some of us came to California, and some of us went to the American South. And my colleagues from high school have become Southerners. And I can tell you, they hate California, they hate Silicon Valley, and they don't want to know that they're in charge of anything. And no, really.
No, I can understand that.
I don't want to go into a religious. I don't want to go into a religious thing, but their ministers are telling them they're not in charge of it.
So your comment is if the solution comes out from fancy Silicon Valley, you have to still get people might be resistant. And
they might even go on a campaign against it.
Yeah. So it's interesting because Francis Crick went to my high school actually in England, in a town called Northampton. Yeah, I mean, we have the anti-vax movement. There's many riposts to many ideas, because we do know medicine is basically a trade-off. Some people it doesn't work, some people it does work, and people have to do that analysis, and that's not usually explained to the masses. I mean, that's really the doctor's job to figure out that discussion. Who else would like to make a comment? Yes, over here.
So my comment is, in order to really cure cancer or any other diseases in whatever number of years, first thing we need to do is remove all the chemicals from everything, including the makeup. Like, I read all the ingredients, most people don't. Like, if you take any product of any of the over-the-counter, which I know that here kids start using since middle school, it has a lot of different things which can cause cancer later on. So it's sodium benzoate, which is in all the store-bought food or frozen food and other things we buy. So I think in order to really get rid of diseases, we need to go to the source of what's causing them. And one of the easiest thing is, I don't know if anyone has done research in all the ingredients in different things we buy, which later on with continuous use can lead to diseases, including cancer. If we handle that part, half of the diseases might get cured or might not even happen. So it might be even better than a vaccine.
So it might be, maybe, this is an AI, this is the AI panel section, but you may have advisors on your watch before you take a bite into something, because
Yes.
realizes that you have a life logger type thing, where you have spectacles, when you're about to not behave yourself or do something that it warns you, a bit like that Terminator.
Yeah,
Okay.
like if you can train the AI to
Okay,
really figure it
one
out.
question or comment. One more question over there.
Hi, I'm Lisa Gashara. I'm a global health student and just interested in listening in today. I was wondering where health equity kind of comes into the conversations and how we're going to be socially responsible, especially including low and middle income countries in the conversations of developing tech-based
So let me just jump
solutions.
in. We have final time. I mean, health equity, the AI part is maybe you don't access to doctors, even if you have access, the copay is ridiculous, the insurance ridiculous. Maybe access the AI system. And Nabil sounds like he wants to comment.
I thought that actually, in a way, relates to the data that we train our model. So if our data is biased, then that's what the model is trained on. So a challenge in terms of having quality data, quality means a lot of things. One of them is to make sure that there is a free of bias. Again, never 100%, but at least if we are conscious of that and try hard to tackle that, I think we get further away.
Yeah, that's the health equity issue at the AI level. Okay, I'm off, right? Is that right? Is it lunchtime or no, someone else? Concluding remarks. Thank you. So we've had some brilliant comments here. I think the general theme here is, AI is here to stay. It's not perfect, but I'm actually more optimistic that in the 2030, 2031, I think we should be aiming bigger. Not curing liver cancer, which is great. Not curing pancreatic cancer. Cure all the cancers all at once and all the diseases, because we now have the tools. We just have to use them.
All right. Thank you, panel. Thank you, Ranjan, and the rest of the panelists.